Connor, by now we've done a few episodes on oligonucleotides, or oligos as they're often known, but there is still so much to talk about.
Yes, and a quick refresher course for our listeners out there, and probably myself as well. Oligos are short strands of DNA or RNA made up of a small number of building blocks called nucleotides. So oligonucleotides, oligos for short.
And today, we're looking at new synthesis technologies, specifically how new synthesis technologies survive contact with manufacturing challenges, regulations, cost pressures, and environmental reality.
The advances in data processing and modeling are phenomenal. It's like having a highly trained manufacturing scientist on your line all of the time. So that's great. It's a brilliant technology.
When we are in this industry, every day, we know we are working on some breakthrough drugs and, life-changing drugs, and that make us, very proud and fulfilled.
Oligos are what matters on today's episode of Discovery Matters.
To start us off, we have Dr. Barry Cassie.
So I've worked in the pharma industry for 25 years. I spent a long time working in manufacturing for GSK, big complex manufacturing site, lots of different processes, really, really interesting place to work. When I say complex, what I mean is challenging. Lots of different challenges, lots of different problems, lots of different opportunities. So that was a very good place to experience lots of things.
After GSK, Barry worked for an innovation center.
So really absolutely laser focused on trying to bring innovations into sort of commercial use, which, from my experience of working in the manufacturing site, is quite challenging. So I've spent my entire career working with technologies, varying kinds, and maybe I'm a bit of a technology nerd.
Aha, a fellow boffin. Don't worry, Barry, you have come to the right place.
The last 5 years, I've really focused in on oligonucleotides, which is a really interesting time to come into oligonucleotides. The whole dynamic of the whole manufacturing challenge has changed dramatically.
So you see, he is basically an industry expert on oligos.
I've been working independently, working with companies of all kinds of sizes, so some quite big companies to, some very small single-person companies, helping them with their technology. And what's nice is to just be able to help them understand what their customer is thinking. So a lot of them have never worked in pharma manufacturing, so they don't really understand the questions that are going through the heads of the people they're trying to get to adopt their technology. So I can really come along and sit and go, well, if I was them, this is what I would be asking. And they'd be sitting going, really? Yeah.
Manufacturing challenges for oligonucleotides are different today than they were in the recent past.
And what are those challenges?
They stem from a small RNA oligotherapy that aims to remove so-called bad cholesterol from the blood. That therapy was first approved in Europe in 2020. 1 year later, it was given the green light by the FDA in the United States for treating heart diseases.
So all the oligonucleotides before inclisiran were rare diseases, hundreds, maybe thousands, not very many patients. And then a heart disease drug comes along. So instead of looking at supplying a few kilos, tens of kilos, you're now talking about tons. And it's a completely different question. It's not just a different challenge. It just opens up things that you would not have considered. Trying to make 10 kilos, there are certain things you just wouldn't even thought about because they would be utterly ridiculous. But making a ton, maybe I'm going to think about doing that. So I think that's where the challenge has changed. And if you look at the pipeline, the number of big patient population drugs coming through is phenomenal. Over 50% of the pipeline is now big patient. You've got Alzheimer's drugs in there. You've got infectious diseases. You've got metabolic. We know how big metabolic drugs are these days.
So if I understand this correctly, the life sciences world is starting to go, okay, oligos can be used for more than just rare diseases. We have something to do about that.
Yes, exactly. And that question was being asked, by some of the people who knew they were going to get landed with the problem. They could see their company's pipeline. They started asking that question sort of 5-6 years ago. So I went, right, we need to do something now, because when these things land, if I'm not giving my company a process that can produce this drug, then those millions and millions of dollars of investment in developing that drug, they're not going to get the return. for that investment if we can't make it. And it's quite unusual to say, can I actually make that amount of this drug? In the sort of traditional small molecule world, that doesn't exist.
Basically, the manufacturing challenge here is moving from something for rare diseases, where patient populations are relatively small, to this heart medication is going to be one of those super drugs. So now drug makers must think about producing millions of doses. And that is why Barry believes right now is an exciting time for oligos.
Technology that's used today to make oligonucleotides has been around in its most fundamental form for a long time, 40 plus years. Do you know why? It's very good. It can let you make any one of a whole range of molecules. It does it quite robustly. You can do the same process to make 100 molecules for screening early on in target development. You can almost use the same process to manufacture in commercial. Developing drugs is all about risk. So if I've got almost zero risk in my manufacturing process, I can go from this tiny scale to a kilo without really having to change an awful lot, and I'm being massively oversimplifying. It takes one of the risks out. I don't have to spend money on it. It doesn't take time. So brilliant. That's great. It's a brilliant technology.
I'm sensing a but here. What's the downside of all of this? Because it can't all be upside.
The problem comes if the technology runs on bed columns, both in synthesis and purification, and they can only be so big, particularly in synthesis because the resin, it swells and things like this, and your column can only be so big. So your maximum batch size, maybe 20 kilos, and it's a complex process. The analytics around it are very complicated. So if I'm making 40 kilos a year, making 2 batches, that's fine. And I want to make a ton at 20 kilos a go. So what's that? 50 batches? I've now introduced an enormous amount of complexity and effort into my manufacturing process. I used to make antibiotics. We had single batches that were 400 kilos. You know what I mean? It's a world of difference. And I think, just the amount of analytical effort going into making a batch makes it making significant volumes on that scale really challenging.
I wonder how much of modern synthesis is chemistry now. Are we really delving into data and software?
So when you're looking at a technology, data is expensive to collect. Not as expensive as it used to be, but if I'm going to put a complex bit of instrumentation or whatever onto a piece of lab equipment or onto a manufacturing line, I've got to know why I'm doing that. What data am I getting out of this? And if you don't collect the right data, then you've got no substance to do anything with. The advances in data processing and modeling are phenomenal. Some of the things that you can do with predictive modeling are excellent, but they all need a basis of data from somewhere. To collect the right data, you need to understand the process fundamentally. So all the kind of any advance in new technologies, you've really got to understand what is happening and what is critical to know what data to collect. Because you could just collect a huge amount of data, very expensive, and it's zero use. So yes, that there are huge advances in data and the potential for things like model predictive control systems. That's like having a highly trained, manufacturing scientist on your line all of the time, looking at all the data in real time and making decisions. It's phenomenal, but you need to have a basis of data to start from. And that's the kind of back end of your development. You start with what's going on, what's my fundamentals, collect me some data, build some models, get me robustness, I understand this process, and then might automate it, might put control systems in. But as a very wise man once said to me, he said, you discover something and you understand it, then you optimize it, then you automate it.
And where are we on that scale with oligonucleotides?
There's some patches of very good technologies, and it's involved in a project recently where using a AI-driven machine learning tool fed data around synthesis and starting to be able to predict synthesis conditions, etc., predict quality from a particular experience of manufacturing, which I was amazed how few experiments. It still took 80 experiments, but that's not many experiments to feed a model that complex. Really impressed by that. Some of the automation of data handling analytics is really good.
Barry stresses that oligonucleotides are not easy to analyze.
You've got a sort of normal analytical technique called HPLC, but you can't really separate very well because you have to use mass spectrometry, which is just super complicated. So some of the vendors are really putting some interesting things into automating that data flow. But then you still have this piece of going, well, someone has to understand what the synthesis is trying to do and what this data is telling me to put the two things together. So people trying to work on that piece as well, trying to really understand, put some intelligence into these automated data processing things, but it's very, very complex.
Should the industry focus on broad drug discovery methods or specialize in automated solutions for specific treatments for things such as heart disease?
That depends.
So pharmaceutical company, their job is discovering and bringing to market new, safe, efficacious medicines. And whilst a lot of them have big manufacturing operations, for them it's a means to an end. You've had your R&D operation develop this fabulous new drug. Look how much better it makes these patients' lives. Great, that's fantastic. You've got this brilliant sort of supply chains, distribution team all set up, ready to go, and they're going, give me the medicine. So you're just a bottleneck. It's interesting that the pharma companies have a hybrid model. They keep some things internal. They externalize some things depending on technology, depending on the age of the product. They will pick and choose. But I think you need specialists to do things. So there are some people who've got fabulous knowledge of control systems, for example. Some of the things that they know can be done, or maybe they're already doing it in oil and gas production. But what they don't understand is what the pharma industry needs. And it's a case of bringing together the right experts, locking them in a dark room for a while. They understand each other. Oh, you can do that.
Staying on the idea of challenges for a while, what about all the sustainability around this?
And that's usually one of the biggest failure modes for a particular synthesis technology. So it could be as simple as maybe you want to change from doing a reaction in one solvent to doing a different solvent because it's a better solvent. But maybe you've got a solvent recovery operation. So it's this huge infrastructure just sitting there that's recovering 90% of the solvent you're using anyway. And if you change solvent, suddenly I change that whole setup and maybe I create a solvent mixture I can't recover anymore. So whilst it might be a great idea to switch into something that's from the creation of that solvent is a more sustainable thing, it creates so many problems in the processing downstream. I can't actually use it efficiently.
So I'm always looking forward to the near future.
Yes, you are.
Fast forwarding 10 years then, what does Barry see as the biggest difference in how medicines are going to be synthesized?
The biggest features of the pharma industry is it moves very slowly. Painfully slowly. And I was going to say that's a problem, but it's not, because these are things going into humans. So it does tend to move incredibly slowly, which can be frustrating, but I think is always bizarrely comforting.
You'll recall when we were all talking about supply chains.
The global pandemic caused drug shortages. Suddenly everyone looked at supply chains and went, why did that happen? It's made people think quite differently about supply chains. So I think what we're seeing is people sort of thinking, how do I become more robust? How do I become more diverse? And new medicines, it's not all about new medicines, because access to drugs is throughout the world is increasing. So all these old medicines, which cost pennies to make, are going to still be made. They're not going to disappear. But the new medicines are much more specific, much more targeted, lower dose. So you've got much smaller, more agile factories. And I think this is what you're seeing, much more of an agility, which again drives some interesting challenges into how you manufacture. If you're churning out the Model T Ford, it's very simple. It's do that again and again and again.
You can have any color as long as it's black.
Exactly. No, but if every car coming off the production line is not just a different color, but it's a different make and model, it's a different challenge. And I think that you've seen a bit of a shift towards that.
Really interesting. And that's the case with personalized medicine, basically, isn't it?
Exactly. Everything coming off the line is a different make and model, but it also has to be scalable in order to be affordable to both sell and purchase.
So before we let Barry go, what about the regulatory side of all of this?
It's an interesting question because generally the regulators are accused of holding back innovation. And whilst they're not completely blameless, it's really the risk profile of doing something new is sometimes just too high. I always say to people, the quickest way to get sacked in a manufacturing organization is to stop supply of a drug or delay it. And delaying a launch. So you've got people going, I've got this great new technology that I can apply to this drug and it'll make it more efficient, quicker, whatever, greener, or the old way of doing it. The old way of doing it, we've been doing for 20 years and I know it works. There's a 10% chance this new way won't work. So what happens if it doesn't work? And regulatory filing gets delayed. or if we don't have the right data in there or whatever, and maybe we go back 6 months or a year. So a drug that's worth, whatever, $500 million, $1 billion a year, that's a very expensive 6 to 12 months. So I think the biggest challenge is that how do you get over this kind of risk of doing something new? And there's the sort of technical, actually physically doing risk, but then there's a regulator turn around saying, yeah, but you haven't provided the right data. And sometimes I just think they could work together a little bit better. And maybe there should be some sort of flexibility. I mean, there are some schemes and things, but there should be a bit more flexibility to say, look, if you come to us with a new technology, it's better.
So basically, you can't apply the same criteria to new technology as you do to the old.
Actually, criteria applied to old and new technology is the same, just the level of confidence in the data is generally higher for established technologies.
There are some good schemes. I think like the FDA have things where you can go and ask questions in advance and things like that, but it still comes down to this, there's a very brave decision somewhere. As my old boss used to say, farmers are raced to be second, because if you're first, you get asked all the difficult questions. And then once the second person comes on, they go, oh, we know about this, that's great. And it's one of the things about collaboration is quite often they're very keen to publish. Because if you turn up to a regulatory filing and you've worked with this academic or a small company and these 2 or 3 other pharma companies, and you've published this data, it's been peer reviewed, that says a lot about your technology or the process or whatever you're doing. It says, this is not us coming up with something random. This is something that's out there, it's been peer reviewed, it's been developed. This is not, you don't need to worry about this.
We've now heard from Dr. Barry Cassie, an industry expert about the challenges of adopting oligos. Now it's time to hear from the other side of the table. I recently chatted with our next guest from WuXi, a company actively investing in new synthesis technologies.
And Dodie, I think, seeing as you've chatted with them so recently, we should just hear the rest of that interview in full.
Can we begin, please, by just getting to know you? So what's your name, what's your job title, and what inspires you to come to work every day?
Sure. My name is William Fong, trained peptide chemist, pretty much working in the CRDMO industry. So right now I'm the VP in WuXi Tides division. So mainly responsible for the peptide oligonucleotide development. The drug we are developing or helping developing with our partner is really changing the life of lots of patients. People suffering the disease, they don't have a good drug. And so I think especially this new field, oligonucleotide field, and this really changed the life of those regular peoples. And this is make my life more fulfilled.
So if these oligonucleotides are so remarkable and will change people's lives, can you describe what makes them different from other biologics that we might be familiar with? And why is this the time for oligonucleotides to come into the spotlight, as it were?
Overall, oligonucleotide and peptide, they are all, we call them new modalities. And in the past, I think the main challenge of oligonucleotide is one, of course, chemistry, one is the delivery. But now the chemistry is working better, but the delivery is still, in some way, it's working, but not perfect yet. Surpass the protein, surpass a lot of the small molecules that only targeting the receptors. So quite a number of undruggable targets in the past now will become available. That's definitely a game changer. And then we believe with the technology continue to improve, this will getting better.
All right, interesting. So you talked about how the delivery has yet to really progress well enough to make oligonucleotides a huge success. And I'm curious, can you tell me more about the challenges in delivery and perhaps in manufacturing? What does this industry need to do to really see oligonucleotides become accessible to more patients?
Right now, most of the antisense siRNA, they are showing number of tangible clinical success, most also especially in the liver-targeted diseases. That's where current delivery is a lot more established. But then beyond that, the overall delivery is still biggest bottleneck, particularly beyond the liver. The people, I think lots of research companies are working on to expand that, but it's so far there's no huge breakthrough. It's more the progress is there, it's steady, but it's incremental. So hopefully by next few years, there will some breakthrough in this area. And in terms of manufacturing, well, the design of molecule is one thing, but then also to deliver a large quantity and consistent quality is another differentiator for this type of complex molecule. For this kind of job to go to the market, to the patient, you need to make sure the product can be manufactured at scale, at quality, and that necessarily to support clinical success.
Sure, but right now, oligonucleotides don't need quantity because they're really focused on rare diseases and patients of rare diseases. So when you talk about needing to be able to deliver at scale and with quality, are you looking ahead to the ability for oligonucleotides to be used for different targets?
There are potential, once approved for one disease area, you may expand further. Like for right now, some of the particular PPMO type of oligonucleotide. This is a special oligo in the sense that design structure change will make the product is much more stable in the body. So the potency will be better. And this is this PMO, the special oligo actually will conjugate typically with a peptide. And that's why it's called a PPMO, it's peptide conjugated PMO. And they actually can expand to different disease function area. But overall, regardless if it's, I guess the quantity compared to small molecule, it's a small, but still it needs certain, in the clinical or in the commercial stage, you still need a certain quantity, and that requires a larger scale manufacturing overall. And it's important from clinical early stage to commercial manufacturing, the product that we delivered need to be maintained a consistent, let's say, impurity profile and quality, etc.
So one of the promise of oligos is quick design, the ability to see, adjust, iterate. So how fast could a program realistically move from design to clinical trials and to actually getting into patients?
Overall, I think compared to traditional, like small molecule, the cycle definitely shorter, faster, because the sequence, genetic sequence, most of them already well known. So pretty much designed based on the sequence, original sequence. With that said, even after the sequence design, then you still need to validate it through traditional clinical trial and testing. So that actually somewhat shortened because some rare disease, the regulatory has special program you can speed up, but the overall still take time.
But I love that. So other branches of science are helping speed progress in oligonucleotides.
Yeah.
That's terrific. So what do you see in terms of looking ahead, let's say 10 years? What do you think success looks like for oligonucleotides?
I think we definitely see, the success of inclisiran, the drug like that, similar like that, actually has been more and more pumping up in the clinical stage. And so hopefully by next 5-10 years, there's a better delivery tool and to treat not just a rare disease, but they'll be able to treat broader disease with broader populations. With help with all the manufacturing tool improvement in the supply chain, we can reduce the manufacturing cost. So the better delivery, scalable manufacturing, and the better cost, hopefully that will benefit the broad population in the long term. So we definitely see in the past 10 years, we see a dramatic improvement in cost reductions overall. So I think that we are confident that another year or 10 years ahead will continuously improve in this direction.
In the same way that during the pandemic, supply chain and mRNA became topics at kitchen table conversations for families that might not be scientific families. Do you think that oligonucleotides will make their way into our everyday conversations, or do they belong at kitchen table conversations? How do you think they will break through into an everyday person's life?
Certainly. So right now, the one popular topic in the peptide field is the GLP-1 type of analog for weight loss. So similar type of drug actually are also in the clinical phase in the oligo field. It's just similar type of medicine, hopefully that will come up in the oligonucleotide field that will become popular.
Can you explain the potential and how that would connect to our everyday lives?
Some of the pipeline, the clinical stage in oligonucleotide, they can deliver consistent weight loss, and they probably don't have to take every day or inject once a week. They can pretty much inject once every half year. And then the current preliminary result shows they don't have the lean muscle loss issues. And we compare to other, let's say, peptide GLP-1 drug. That's a huge benefit for the patient.
Yeah, exactly.
Yeah.
That's just clinical trials, so that's not anything to count on, but that is an example of potential benefits in a patient scenario.
Yeah.
I just want to ask William about what excites you about, you are focused on oligonucleotides. That is your day in, day out job. And when you talk to your neighbors about what you do at work, what brings a smile to your face about what you are doing at work?
I think the field that we are in is really different than other field. For us, we are really in a tangible area that whatever medicine that being developed through our organizations and group, that really can transform the life of the patients. I have some relatives, they are sick, but then they don't have good drug in the market. Even they're willing to pay whatever the cost, but there's no drug. But then if there's a drug in the market, quite often it's the saving of life in other way. So I think that when we are in this industry, every day, we know we are working on some breakthrough drugs and, life-changing drugs, and that make us, very proud and fulfilled. For us as a scientist, we believe in science. Together, we just in this field will definitely further help improve the process faster to the market. It will be a trend, and with this kind of new area of innovative new drugs, I think for the longer term, life will become better. And quite often right now, sometimes we can look at the oncology side. A lot of people who had a tumor or whatever, it's treatable. And I think in the long term, this will be further improved. We are optimistic in this field.
So today we learned that new oligosynthesis technologies aren't just about innovation. They have to survive real-world manufacturing challenges, cost and regulatory pressures if they are to make it to market and have an impact on patients.
And scaling from rare disease treatments to massive populations, that completely changes the game.
And it also sounds to me like data and automation are powerful, but only if you really, 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 really understand the fundamentals first.
Even then, the biggest barrier can be risk. Companies traditionally stick with what works because failure costs a lot.
Which means progress depends on balancing innovation with reliability, bringing the right experts together, and ultimately, the future of oligos isn't just better science, it's smarter, scalable, and more collaborative manufacturing.
Speaking of smarter, let's see what we have learned this week. It is time for Every Day is a School Day. What you got, Connor?
It's kind of a little bit mad because it's oligos too, in a way. But do you ever look at like salmon and think, oh, that's the future of my face and my computer? I bet
no.
you don't. Right, no. But
I do think, though, eating salmon is going to help my brain. I do think that.
It certainly will. Well, I want to talk about salmon milt, MILT, basically fish sperm or the fluid surrounding fish sperm. It is the ultimate liquid gold, believe it or not. And salmon DNA being 97% identical to human DNA, woo, I'm a fish, because it's super compatible with our bodies. Doctors are using it to heal us. If you ever heard of salmon DNA facials or PDRN treatments, that is salmon milk, and it is packed with polynucleotides, short chains of DNA or oligos that act like biomessengers, and they tell your cells to stop slacking, boost collagen, repair skin tissue from the inside out. So it's like oligo power from the salmon. Extraordinary.
That is.
Absolutely mental. It gets weirder because the DNA in milk is also a natural polymer. So researchers are using it to create, get this, biodegradable plastics. They even found out that the phosphate in this DNA is a magnet for rare earth metals. That means, get this, we can use salmon milk to recycle the neodymium used in your smartphone and EV batteries. So the next time you see a salmon, remember, it might not just be dinner. It's a source of life-saving medicine, a skincare miracle maybe, and maybe even the reason your next phone will be more eco-friendly.
Or your EV. Yeah.
Or your EV, indeed. What about you, Dodie? What have you learned?
My story is also about gene therapy, but this is about what we see or what we don't see. I found this on Business Insider, which is always a good site for, a quick read on interesting things going on in the world. So scientists reported strong results from a clinical trial testing a gene therapy injection for a common cause of age-related blindness. And this therapy delivers a gene directly into retinal cells. And after 1 year, the patients in this trial showed a significantly slower loss of vision without side effects. So striking in that this is not correcting a single broken gene, but gently rebalancing an entire gene network. Not a cure yet, but certainly a big step toward long-lasting treatments for vision loss from a single gene therapy shot. Our producer, mixer, and sound magician is Mr. Tom Henley. My name is Dodie Axelson.
And my name is Connor McCaffney. Please rate us wherever you get your podcasts from. For some reason, in mysterious ways, it helps the algorithm understand who we are and what we do. We'll see you next time on Discovery Matters.